Okay, here is Unit 1, Part 2 of the essay questions. So we're going to start with resting membrane potential and how the cell membrane is established. So resting membrane potential is the equivalence point of the electrochemical gradient of a cell, meaning the concentration of the ions in and out of the cell are equalizing each other out, and same with the charges of those ions as well. So that typically means that the inside or outside of the cell can be more positive or more negative. Usually the inside of the cell is more negative, and that is due to things such as proteins inside of the cell that are negatively charged, or things that are more found inside the cell, such as potassium. If they were to leave the cell, that causes the cell to be more negative in nature, since potassium is positive and it's leaving. Same as opposite is true. The outside of the cell is typically more positive compared to the inside of the cell because of ions such as sodium, calcium, and chloride. And those guys typically have a higher gradient going into the cell. So the membrane potential is just based off of the Nernst equation, which is negative 61.5 over the charge of the specific ion in question, times the log of the concentration of inside the cell, the ions inside the cell, over ions outside the cell. So once you determine that the Nernst equation potential, use that to help you understand how polar or non-polar a cell is, or an ion is, compared to the cell and its surroundings. And that kind of leads you to the idea of action potentials. So action potentials are the way cells, such as like neurons, for examples, fire and create a wave of electricity or gradient through itself. And typically for neurons, this passes through from one cell neuron to another. How this happens is, so the cell includes a bunch of different kinds of channels, as discussed before. And these different channels allows for these ions to either pass into or out of the cell. Some of them are naturally permeable. Some of them are not, and they require energy to be able to move across those membranes. And so the cell typically rests at the resting membrane potential, like we discussed. For example, for a neuron, that would be negative 70 millivolts. And that's just, even though there are things like leak channels for potassiums that kind of go out, sometimes it's at an equal rate of coming in as well due to other things. But in a hypothetical situation, well, let's say this neuron has a little spike into its polarity, that some sodium would to go into the cell. This makes the cell more positive in nature. It is called depolarization. And the way those gradient potentials could happen is many and far in between, but once they happen and depolarize the cell high enough, it can reach something called a threshold. And the threshold is what keeps the channels closed or open. If you go above the threshold of a cell, then that in turn opens all the activation gates of those different ion channels. and the gradient of all those ions are going to move wherever they want to move. Once it hits that threshold, a lot of sodium rushes into the cell, making it depolarized really quickly. And because the cell wants to reach a homeostasis or equilibrium, it will open potassium ion channels, voltage ion channels that are going to release those positive ions of potassium out of the cell, trying to equalize the sodium and bring it back to that -70 for those neurons. And so in turn, that starts to repolarize the cell back down below its threshold where it wants to be. And this action for the voltage potassium channels are actually really slow. The inactivation gate can continue to be open until even past the point of resting membrane potential of -70. So once it goes below or more negative compared to what the cell normally is at rest, that is considered hyperpolarization. But eventually, once those gates are finally closed, the leakage gates that exist for the potassium will allow some to flow back in so that the cell can reach that resting point again. And so I guess there's not technically a term for that, but you can say that is technically depolarization, but that's just the cell going back to its resting state. So during the time of those sodium ions rushing in and then the potassium rushing out to reach that equilibrium again, there are refractory periods, which basically means that there are periods within that time frame where there cannot be another period of those events happening at the same time as it already is happening, which actually is called action potential. So the action potential can't really happen again during a refractory period. We have a period called absolute refractory period. This is during that big spike of depolarization to repolarization of the cell. And then there is relative refractory period, which is that point where the voltage potassium ion gates are still closing, but is still quite open enough, and the cell hyperpolarizes. So that is the relative refractory period. Another action potential cannot happen during either of those times. Let's see. So yeah, graded potential is anytime a depolarization event happens but doesn't quite reach threshold and creates the action potential. An action potential is any time that the threshold is exceeded and the gates of all the ion channels open. So for a cell, like a neuron, you also have to think about what its job is trying to do. It's trying to send those ions down the length of a neuron and in turns interact with the neuron or the cells that are next to it. In order to do so, we have to talk about capacitance and length constant. So capacitance is basically how permeable an ion is to a cell. Now, this depends on many things, of course, but such as how many gates, ion channel gates that ion has for the cell, or how much surface area this cell has, right? How permeable is it to the cell in reference to its gradient? The cell, like I mentioned earlier, is more negative on the inside and relatively more positive on the outside of the cell. When it comes to neurons, those charges typically attract each other. Since opposites attract on that membrane, those positive and negative charges are trying to pull towards each other. but sometimes it's not perfect, and let's say a potassium ion leaks out of the cell, and those extra positive ions are trying to repel the other positive ions around it. There's also ways where the cell, when it does have an action potential event, it is kind of interfered a little bit by things around it, such as those native proteins that are negatively charged proteins, that are in the cell, right? If other negatives are repelling each other, it causes different actions, and thus it makes it harder for those signals to be sent across through a neuron. So that's kind of a little bit about capacitance. We have length constant as well. So the length constant is the resistance within the cell. So let's say the cell's membrane has these charges, both positive and negative. When you have myelin included, that is just fatty substance on the outside of the axon of a neuron. It kind of separates those charges from being so close to each other, and thus it creates less of a resistance to those signals from the action potential that tries to run down the length of the neuron. But the more of those charges you have on the membrane, the more it can actually affect or slow the signals of action potentials. So the myelin in this case will try to separate the charges and try to make it so that there's less resistance to those signals moving down the neuron. And another set of resistance we have is something called internal resistance. I mentioned before, it's the negatively charged proteins that exist, or even other ions that are floating around in the cell. Depending on its charge, it could also affect the signal of the action potential moving down its neuron. Thus, it requires more action potentials to occur within the cell in order to achieve its intended purpose. And so myelin doesn't really have an effect on the internal resistance, just because it is typically on the outside of the cell. But the length constant will help us to understand that once we have less membrane resistance, we have a decrease in length constant. But if we have an increase in the membrane resistance, then we have an increase in length constant. Then the opposite is true with internal resistance. If we have a greater internal resistance, we have less of a length constant. And if we have less of internal resistance, then we have greater length constant occurring in the cell. I believe that is all for this part of the unit. And yeah, see you next time.